Good morning. I'm David Reby. I'm a professor of ethology at the équipe de neuroethologie sensorielle at the University of Saint-Étienne in France. I'm going to talk today about the evolution of size communication in mammal vocalizations. Why size? Well, body size, is a very important factor that is known to affect the outcomes of male competition and also of female choice. And we also know that selection for large body size in males results in size sexual dimorphism, especially in polygynous species. As a consequence, size communication is expected to be an important function of sexually selected vocalizations. But before I start, let me take you on a tour around the globe to see the effect of sexual selection on male sexual signals. And to do this, I'm going to follow the example, look at the example of polygynous deer. Polygynous deer are a group of species that are present around the world, and they produce rutting calls, which are called roars, groans, bellows, howls, growls, yodels, moans, or bugles. And the diversity of these vernacular names, in fact, reflects a very strong bioacoustic diversity. And I'm going to quickly play these vocalizations. So this is the European red deer. Not the prettiest vocalization, I think. This is the Spanish red deer, a subspecies, maybe slightly more singing. This one, which sounds quite big, is the Corsican red deer. It's actually one of the smallest. This one, which also sounds quite big, is from a different genus. That is the fallow deer, which originates from Mesopotamia. Now I'm going to play the barasinger. It's quite something. The Per David's deer. The Taiwanese sika. The Timor sika. The Japanese sika deer. And finally, the biggest of all, the American elk, or also known as wapiti. So in this one is quite interesting, and we'll come back to it later. Now, how can we investigate the origins and the function of such an incredible acoustic diversity? To do this, well, I use an approach that combines three levels of investigation. First, anatomical investigation, where we perform dissections, but also we use imaging techniques in order to get biometric measures. And the investigation of the functional anatomy of these species, of their vocal organs, enables us to make predictions about which parameters in the vocalizations are going to encode information. Then, in order to verify these hypotheses, we obviously record live animals, and we perform acoustic analysis where we extract specific spectral components which are guided, obviously the choice of these components are guided by our understanding of vocal production as informed by our anatomical investigations. And once we find correlations between traits, size, for example, and for example, frequency components, then we can do playback experiments where we test whether animals are capable of perceiving and using the information contained in these components. And to do this, we as we'll see, we have we can access very powerful synthesis and resynthesis tools. Now, to do this approach, I use a methodological framework which is called the source filter theory of voice production. Now, according to this paradigm, the voice of any vertebrate, terrestrial vertebrate, is produced in a two-stage process. The first stage takes place at the level of the source, which is the larynx, where as air is forced out of the lungs through the glottis, it causes vocal folds to vibrate, which is going to generate what we call a glottal wave, which is composed of a fundamental frequency and a series of harmonics. Now, the fundamental frequency is responsible for the pitch of the voice and is determined by the length of the vocal folds. So, if I'm going to play you a red deer roar, which produced by the larynx only. Imagine a red deer larynx without the head and neck of the deer on top of it. So this is what the source signal would sound like, and you can hear a pitch, a fundamental frequency, and an intonation, that is the variation of the pitch. But then, fortunately, this signal is filtered through the vocal tract of the animal, through what we call the supralaryngeal cavities of the animal, and this filtering process is going to confer the signal its natural resonances, which are going to shape the frequency envelope of the glottal wave and creating broadband frequency peaks, which are called the formant frequencies. Now you can see these formant frequencies here, F1, F2, F3, F4, and F5. And the resulting signal, once the glottal wave is filtered, sounds a lot more like a red deer roar. Now, one key assumption of the source filter theory is that the source, that is the pitch, is independent of the filter, that is the vocal tract resonances. So there is no feedback of the filter on the source. An animal can produce potentially a signal with any resonances independently of the value of the pitch and vice versa. So both, to say it perhaps better, both source and filter related features can independently encode information about the emitter, about it, for example, its stature, its condition, or its motivational state. So understanding how these features are produced can lead us to make informed predictions about what information they contain. And there are two obvious candidates for size communication in vertebrate vocalizations. The first one is the fundamental frequency. Indeed, larger animals should have longer vocal folds and thus have a lower fundamental frequency, a lower pitch voice. But also the second one are the formant frequencies, as larger animals should have longer resonators, longer vocal tract, and their lower vocal tract resonances or formants, and therefore a deeper timbra. First, I'm going to talk about size at the source. So we're going to look at the potential for the fundamental frequency to encode size information. Now, I'll go very quickly over this equation here, but it relates the fundamental frequency of vocalizations to the length of the vocal fold that of the animal that produces it. So according to this equation, the fundamental frequency should be inversely correlated to the length of the vocal fold. The longer the vocal folds, the lower the pitch of the vocalization. If we look at interspecific variation, and if we compare the fundamental frequency, the pitch of the voice of animals of a wide range of weights, we find that this relationship, this inverse relationship, is clearly rather strong, with elephants having the vocalizations which are infrasonic, and at the other end of the spectrum, bats having producing ultrasonic vocalizations. Now, even though this regression line is quite strong, there are clear outliers. For example, koalas, fallow deer have lower pitch vocalizations than expected for their body size. And on the contrary, the American elk and the horse, domestic horse, have a much higher pitch vocalization than expected for their body sizes. And we'll come back to these later. So, for example, koalas produce bellows. I'm going to play one of these. think that's enough. And these bellows are much lower, have a much lower pitch than expected for these from the size of these relatively small animals. And in fact, in order to produce these very low fundamental frequency, these very low vibration, they do not use their vocal folds, which are located in the larynx and are rather small, but they have evolved a secondary set of vocal folds at the entrance of the nasal cavity, which are called velar vocal folds because they involve the velum, which is the soft palate of the koala. So in this species, selection to sound perhaps bigger or maybe more dominant by producing very low pitch vocalizations has led to not to longer vocal folds, but to the evolution of a secondary set of vocal folds in the nasal cavity. At the other end of the spectrum, we have a species that produce that are very large but produce very high pitch fundamental frequencies. I'll play the American elk bugle again. And this is a whinny from a domestic horse. Now, how can these very large animals produce such high-pitched vocalization despite having large larynges and long vocal folds? Well, in both cases, the production involves biphonation, and the high-frequency component does not necessarily involve vocal fold vibration, at least in the wapiti, and I'll come back to this at the end of the talk. We suspect that the high-pitch component is a whistle rather than produced by vibration of the vocal folds. So in both cases, in order to escape the allometric principle that relates body size to pitch, animals appear to be evolving a more complex mode of voice production. And such similar anatomical adaptations, but this time involving directly the larynx and the vocal folds, are actually found in many species. For example, fleshy vocal pads that slow down the vibration of the vocal folds have been identified in roaring cats, in Mongolian and goited gazelles, or also saiga antelopes. And hypertrophied larynges are found in howler monkeys and also in hammer-headed bats, which basically in these bats, the larynx is so big that it occupies most of the chest cavity, which has led some colleagues to call these animals flying larynges. Now, if we look again at interspecific variation of F0 across an even wider range of mammals, we can see that the relationship is even stronger once we exclude the animals that have evolved these adaptations, either an additional sound source, as we've seen in horses, wapiti or koalas, or an enlarged larynx or thickened vocal folds. So once we exclude these specific adaptations, then we find an even stronger correlation. Now, if we go back to polygynous deer, there is, in fact, very interesting variation in F0 between species. And in some species, like the fallow deer or the Corsican deer, and I'll play these briefly. This is the fallow deer. This is the Corsican deer. These animals are relatively small in size, but they have a very low fundamental frequencies, which are supported by a very large larynx and thick vocal folds. Now, interestingly, in both subspecies and subspecies of deer, there is a very strong sexual dimorphism in the length of the vocal folds and also in the fundamental frequency. So it's really suggests that in this species there is strong selection in males to produce a lower pitch than expected for their body size. Red deer, in contrast, are of relatively large size, but the pitch of their vocalizations is in fact quite high, even though they have a large larynx. There is some sexual dimorphism in vocal fold length and very limited sexual dimorphism in fundamental frequency, which actually we'll see disappear if we correct for size differences between the sexes. Finally, Sika deer have produced a call which has a very high fundamental frequency, which is supported by a very small larynx and very thin vocal folds. And now in this species, there is no sexual dimorphism in vocal fold length and in fundamental frequency. Here, this is just to illustrate the point I was making. These are the sections of the larynx where you can see the vocal folds here of male and female fallow deer, and that you can see that in the fallow deer, the larynx extends considerably ventrally, allowing for the development of very long vocal folds in adult males. In the contrast, the Sika deer larynx is very similar to the fallow deer female larynx, and is not sexually dimorphic, as it is the same in males as it is in females. In fact, in Sika males, the larynx, the body appears to be growing during the successive puberties. All the body grows, so the species is sexually dimorphic in terms of body size and other traits, but the larynx seems to be immune to the effect of sexual hormones and remains female-like in males. If we look at how the length of the vocal fold increases in males and females of the species, we can see that in fallow deer, the length of the vocal folds grows disproportionately with age in males than in females. We have the similar but less pronounced differential growth in red deer, but in contrast, in Sika deer, the male larynx has very similar dimension to the female larynxes as the animals grow. And perhaps more interestingly, if we plot the length of the vocal folds against the length of the jaw, then we see that the larynx, the vocal folds, grow disproportionately larger in fallow deer males than in females, but not in red deer or in Sika deer. And in fact, if we correct for age, body size does not predict vocal fold length in either species, suggesting that vocal fold length and, as a consequence, the pitch of the voice is not going to be an indicator of body size outside of age-related variation in size. And generally, F0, the fundamental frequency or the pitch of the vocalizations, is a very poor indicator of inter-individual size variation within age and sex classes in mammals. So body size does not predict F0 in red deer stags among adult males, in fallow deer bucks, and in human males, it only explains 2% of the variance, which makes it a very unreliable cue to size. And interestingly, if we correlate the fundamental frequency of species with the extent of their size sexual dimorphism, we find that males from more sexually dimorphic species do not have a significantly lower fundamental frequency than expected for their body size. And this means that once we correct for sex differences between males and females, the males do not have disproportionately lower F0s in more sexually dimorphic species. And this suggests that selection for low fundamental frequency is decoupled from size communication in this species. Yet at least in human listeners, having a lower fundamental frequency is erroneously associated with larger body size. Men with lower pitch voices sound bigger to human listeners. So what about other species? Where to test the possible function of F0, we have an amazing tool which is called solar resynthesis, and which allows us to modify the pitch of the voice or the pitch of the vocalizations independently of all other parameters. So here I'm going to play 3 stimuli, 3 red deer rolls, an original one, and 2 rolls where the pitch has been raised by 30 Hz or lowered by 70 Hz. Original, raised pitch, and lowered pitch. So here the pitch can be raised or lowered independently of the duration of the roll, of the intonation of the roll, or of the formant frequencies of the vocal tract resonances. And then we can use these vocalizations in playback experiments to see if animals appear to pay attention to a variation in fundamental frequency. So we did, Maxime Garcia and I conducted these playback experiments, and we found that when we played back these different pitch variants to stags that were holding harems, we did this in Richmond Park in London, there was no significant effect on the response of the males. So it doesn't look like stags pay much attention to the fundamental frequency of the roll. So the fundamental frequency in these species does not seem to have a fundamental effect of a strong effect in male competition. In contrast, in fallow deer, which interestingly is the species where F0 is very strongly sexually dimorphic, experiments conducted by Ben Pitcher and Alan McElligot's group found that when the fundamental frequency was lowered, males paid more attention, and suggesting that they felt that these signals were more threatening. If we look now at female preferences, playback experiments conducted with Kyle McComb and Ben Charlton found that females preferred roles that surprisingly had higher than expected, higher than average fundamental frequencies. So using solar experiments, we played back rolls with either raised or lowered pitch to females that were in estrus, and we found that females were more attracted to the higher pitch variant than the lower pitch variant, which was quite a surprise. And in fact, while this suggests that the fundamental frequency may be mostly driven by a female preference in red deer, it appears to be driven in the opposite direction as expected. And interestingly, maybe in support of this observation, previous work has found that the pitch of the rolls is positively correlated rather than negatively correlated with reproductive success in a wide-ranging red deer stags. So I'm going to conclude on the relationship between the source, the fundamental frequency, and size. We've seen that the fundamental frequency broadly co-varies with body size at interspecific level. Smaller species produce higher pitch vocalization than larger species. And we've also seen that the fundamental frequency reflects size differences between sex and age classes. Where males are larger than females, then tend to produce lower fundamental frequency calls. And obviously, larger adults tend to produce lower fundamental frequency calls than smaller infants or offspring, youngs. However, the fundamental frequency is a poor indicator, in all species studies so far, is a poor indicator of individual differences in body size in same-sex adults. So in low fundamental frequency species, F0 may be an indicator of dominance, possibly mediated by the effect of androgens on the development of the larynx, as well as on dominance. Whereas in high pitch species, the fundamental frequency may correlate with other fitness traits. For example, animals with higher stamina or strength may have either higher lung capacity or a higher capacity to produce a strong subglottal pressure, which is correlated with high pitch in vocalizations. Now I'm going to switch to the potential for filter-related components to convey information on body size. Now, here too there is a relationship between the length of the vocal tract, the length of the resonator, mostly the oral cavity in mammals, and the formant frequencies and the vocal tract resonances. The formant frequency spacing, that is the spacing between any two consecutive formants, is inversely related to the length of the vocal tract. So animals with longer vocal tracts are expected to have lower formant frequencies and overall a lower fundamental frequency, formant frequency spacing. Now, if we look at this in mammal vocalizations, we see that formant frequencies here, this is here a red deer hind contact call, and this is a spectrogram of a roe deer buck bark. Formant frequencies labeled F1, F2, F3, F4, F5, and so on, are horizontal features which are static and which are relatively evenly spaced. Now, if we measure the spacing between the formants and we apply the equation that I just presented, then we can estimate the length of the vocal tract of the animal that produced these vocalizations. And indeed, we find that for a red deer hind contact call, the vocal tract is estimated to around 28 centimeters, which broadly corresponds to the size of the skull of a red deer hind. In the much smaller roe deer buck, we find that formant frequencies spacing is around 800 hertz, which leads to an estimation of a vocal tract of about 20 centimeters, which corresponds to the size of a red deer buck head. So in this species, the length of the vocal tract is mostly constrained by the size of the skull and remains constant during vocalizing. As a consequence, we expect formant spacing to provide a good indicator of body size. And indeed, if we look at interspecific variation in formant frequency spacing, we can see that here too there is a nice inverse relationship between the size of the species and the average formant frequency spacing in the vocalizations that they produce. Again, elephants are the largest on this data set and produce the lowest formant frequencies, whereas surrogates tend to produce the highest formant frequency spacing. We do not see the smallest species here, such as mice, because their fundamental frequency is so high that formants are not resolved and measurable in their vocalizations. And or have not been studied as a consequence of this. So we can conclude that formant frequency spacing is indeed negatively correlated with body size across species at interspecific level. Now, within age and sex classes, formant frequency spacing is also negatively correlated with body size in human speech and in giant panda bleats. And playback experiments have shown that listeners attend to this information in both species. However, some species have evolved adaptations that enable them to modulate the length of their vocal tract and therefore to modulate their formants, especially to lower their formant frequencies. And a classical example of this is found in red deer stags that are capable of extending their vocal tract when they produce their roars. So red deer stags have an Adam's apple like human males, And if you look at this lump in the stag's throat, then you will see that as the stag produces its roar, it lowers its larynx down the sternum. As it does so, it extends the length of its vocal tract, and as a consequence, the formants in the vocalizations are lowered until they reach this minima plateau here, to which I'll get back in a second. But first, let's look at this video. You can see that the stag, as it produces its roar, makes an effort of extending the length of its vocal tract by stretching its head, raising its head, and at the same time lowering its larynx. And in fact, I won't play this video because it says cannot play media here for some reason, but fallow deer bucks also lower their larynx when they produce their vocalizations. So it's not just red deer, but we'll see many other species that develop this independently, this ability. If we look at the anatomy of the vocal apparatus, we can see by here doing a CT scan of a dead specimen, we can see that in the resting position, the larynx, which is typically in mammal quite high in the neck, opening inside the nasal cavity, is already low in resting position thanks to thyrohyoid ligaments, which are elastic in this species rather than an articulation in other mammalian species. And the elasticity of these ligaments combined with strong sternothyroid muscles enable the animal to to lower the larynx and therefore extend the vocal tract during vocalizations. This is just a mid-sagittal slice that allows us to visualize the vocal tract cavity, the oral cavity, the nasal cavities, separated by an elongated velum in the resting and extended positions. Now, interestingly, as I mentioned, when the vocal tract is fully extended, the length of the vocal tract is correlated with the length of the neck of the animal. And as such, formant frequencies produced during this maximal elongation are likely to correlate with the body size of the animal, which was verified in our study of roars on the Isle of Rum, where we found that the apparent vocal tract length estimated from the formant of the roars of the stags could be predicted by the body weight of the animal that produced it. And this result has been, in fact, replicated in fallow deer bucks, where similarly the minimum formant spacing produced during the groan is negatively correlated with the body size of the adult buck. So this leads us to talk about honesty in an exaggerated trait. Indeed, in both species, even though we can infer that gradual cheating on acoustic size by vocal tract extension has probably led to the evolution of a descended and mobile larynx, the presence of a further anatomical constraint, that is, the sternal attachment of the muscles that are responsible for lowering the larynx, has reintroduced honesty in the signal. Now, in order to test the function of formants as an indicator of body size, we can do playback experiments, again using solar resynthesis, which this time can be used not to resynthesize the pitch of the roars, but to resynthesize the spacing of the formants. And here I'm going to play 3 roars where the pitch and the durations are constant, are unchanged, but where the formant frequencies have been raised by 30% or lowered by 30% in order to mimic, in this case, quite drastic size variation. So I think we agree that the last one sounds a lot bigger than the one in the middle. Now, when we played back a similar variance, resynthesized variance to red deer stags on the Isle of Rum, we find that stags reacted much more strongly to the playbacks where the formants were lowered in order to simulate large competitors. And the same was found in fallow deer, where clearly males responded more strongly to the playbacks of groans, this time where the formants had been lowered to simulate larger bucks. In terms of female preferences, we did similar playback experiment as we did for the pitch of the roar to red deer females in estrus, and we find that the females preferred the roars, again with lower formants, and particularly outside the natural range of variation. So females in estrus were spent more time and were more attracted to the roars of stags where the formants had been simulated, had been lowered to simulate larger males. And in fact, this adaptation, having a low larynx and the ability to lower the larynx further during vocalizing, is not just found in red deer and in fallow deer, but also in the North American elk. You can see the larynx go down here and back up, but also in the Corsican deer. Okay, so all these species show a convergent evolution of having a descended at rest and also a movable larynx. But not this trait is not found in all species, at least not in this form. In the Père David's deer, you can see that the larynx is in a high position at rest and only partially lowered during vocalizations. And finally, in sicker deer, the larynx is which we've seen is very small in adult males, is neither descended nor mobile. So this variation between a closely related species suggests that very different selection pressures operate on their vocal communication system, probably as a consequence of their the type of reproductive system. But species from several groups of mammals have also independently evolved elongated vocal tracts. Males can produce abnormally low fundamental frequency spacing for the much lower than expected for the body size by extending the vocal tract using either descended and mobile larynges in antelopes and koalas or lions, but also additional resonators as in howler monkeys, or nasal proboscises as in the proboscis deer, proboscis monkey, or as in the elephant. And in fact, again, if we plot the residual formant frequency spacing, that is the difference between the formant frequency spacing in the vocalization and that predicted from the size of the male that produced the call, again, the species size, sexual size dimorphism, we found that unlike in the case of F0, males of more sexually dimorphic species produce sexual calls with more disproportionately low formant. This suggests that in species where the male is sexually dimorphic in terms of body size, which suggests that males have been selected for having a larger size, males also tend to have evolved adaptation that enable them to sound bigger than they actually are. So that suggests that size communication drives the evolution of these anatomical adaptations for low formants, and its correlates size exaggeration. I don't think I have time to talk about nasalizations, but I'll mention it very briefly to say that we use CT scans to predict the resonances produced by either just the oral cavity or the nasal cavity or both. And we found that, for example, in fallow deer, the nasal cavity is very likely to be involved in the production of of vocal tract resonance, or the production of the groans. And one possible one effect of this is that it adds formants to the vocalization, which could also contribute to making males sound bigger than they actually are, but by adding nasal formants to their oral formants. So in order to conclude on the role of the filter and vocal tract resonances in the expression and communication of body size, we've seen that the formant frequency spacing encodes body size at interspecific level, but also reflects size differences between sex and age classes, and that unlike fundamental frequency, it is a reliable indicator of body size within sex and within age classes. We've also seen that males of several sexually dimorphic species, sexually dimorphic for body size, have evolved adaptation that enable them to exaggerate the perceived size by increasing formant density. But we've seen that in many of these species, the signal has a potential to remain honest due to further anatomical constraints. And in the case of the red deer, because of the attachments of the muscles that are responsible for this vocal tract extension. Now, before I conclude, I would just briefly like to talk about an interesting interplay between the source and the filter. It is in order to highlight formant frequencies, in order for formants to be salient, perceptually salient, the fundamental frequency has to be quite low. It has to convey a high spectral density to the vocalizations. And in fact, formants, for example, in the very high-pitched seeker moans, formants are not visible and not resolved, and they're thus not perceptible. And interestingly, too, for a given vocal fold length, lower pitch vocalization have lower amplitude than higher pitch vocalizations. In fact, even if we compare species, fallow deer groans are very low pitch, but are much softer than the seeker deer moans, which are very high pitch. Thus, there appears to be a trade-off between having a high amplitude, which is facilitated by having a high pitch, and having an efficient size communication, which requires the expression of formant frequencies, as facilitated by also having a low fundamental frequency. Interestingly, some species, for example, the Spanish red deer, produce two types of roars. One which is low pitch and highlights the formants, and it's rather soft and doesn't propagate very far. And one which is high pitch, has a very high amplitude, but does not resolve formant frequencies. So some species seem to be capable of going around this trade-off between size, expression, and amplitude. And in fact, the American wapiti appears to have found a way to sound both large, loud, and sexy at the same time. I will play again this wapiti bugle. And if you pay attention, you can hear both a red deer roar, which, if you look at the spectrogram here, has a fundamental frequency which is quite low, and is perfectly consistent with the length of the vocal folds of these animals. But at the same time, a very high pitch whistle, which is very seeker-like, and which we believe is produced in the nasopharynx. So by using biphonation, the wapiti is capable of producing a very loud, high-pitch whistle that propagates very far because it is emitted at a very high amplitude, while producing a low fundamental frequency whose harmonics are very close and provide high spectral density, resolving the formants, which you can see the animal lowers during the vocalization, probably in order to exaggerate, to maximize the expression of its body size. So I'll conclude with this, and I'll thank my collaborator, and I thank you very much for your attention. is humans, where in human males at puberty, under the effects of androgens, the vocal folds increase. It gives us our Adam's apple, and it means that the pitch drops considerably, or almost to half of its original value. Now, in terms of did we take this into consideration? Yes, we obviously did, but I'm not sure what species the question refers to. And since I talked about quite a few species, it's difficult to understand. As I said, to answer the question properly, as I said, I mean, most of the work, when I mention adult animals, I mean over 6-7 years old for a deer. And in terms of adult, sexual maturity, over, say 14, 15 years old or in that range for humans. Yeah.
Okay, cool.
So I hope this answers the question. But yes, obviously, the pitch varies with age as much as it varies with body size, depending on the species, obviously.
Yeah, I think it does answer the question. Thank you. And then that was posed by Sasha Dime. So you can also go on. That was to the question and answer session, and also type answers in there. So if you want to sort of elaborate on that at all, then that's the space to do so. Okay, I'll move on to the next question, which says, interesting stuff, David. Any idea if the hearing ability of animals also scales with their body size? And or the sounds they produce. So, e.g., those ones off the curve, do koalas hear lower frequency than expected? And if so, would this make animals of certain size more susceptible to anthropogenic disturbance, for example, city noise?
Okay, there's quite a lot of questions.
Quite a lot in that question.
I would say that, while the hearing sensitivity may be tuned to the sounds, the species-specific sounds, that's a possibility that could be investigated, but we have no evidence on this, at least in deer, for example. Megan Wyman, who did a postdoc with me a few years ago, did scan the auditory canal of all these deer species that we studied, and we're hoping someday to see whether the dimensions of this canal are match, for an optimal transmission of the frequencies which are species specific. In terms of the functioning of the cochlea, I don't think that we expect that any allometric relationship that is going to fine-tune the hearing to the species vocalization. But I'm not a specialist of hearing, and I don't want to give a definite answer on this, because it's probably not sufficiently well informed. But there is no evidence of this. And in terms of the relationship with anthropogenic noise, I think one thing that is quite important to realize is that when I say that the pitch is low, it doesn't mean that the vocalization uses a narrow, low spectrum of frequencies. The vocalizations have harmonics, and the harmonics, for example, in the fallow deer, the pitch is very low at 30 hertz, but there is energy all the way to 10,000 hertz and more. So it's important not to confuse the pitch, which can be extremely low, with the bandwidth of the vocalization, which can be extremely wide. So having said that, I think the second part of my answer is that the bandwidth of anthropogenic noise is also very, very wide, and I cannot imagine a direct relationship between hearing sensitivity being tuned to the pitch of the vocalization, being tuned to the size of the animal, and this having a major effect on the response of animals to anthropogenic noise in terms of hearing. In terms of their vocalizations being in or out of the range of anthropogenic noise, it really depends on what noise we're talking about. I know it's kind of fudging but it's there's lots to there's probably an hour to discuss.
There's a lot. It's very complicated.
Parameters involved in this. Yes.
Yeah. Gosh. Okay, I've just, hang on a second. Okay, so I think this is a related question, but any obvious effects of the ecology or environment on the evolution of the F0 or the delta frequency? So for example, in forest dwelling deer versus open plains deer, and anything to do with resonance there?
This is a really good and really interesting question, and I'm afraid I don't have a definite answer to this because again it's very complex. I mean, it would be tempting to correlate the frequencies that the animals use as we record them now with then the question is with which habitat, with the habitat in which they evolved, and it's very difficult to get very definite answers as to which habitat did these subspecies or species of deer evolved in, and then the other problem is that, for example, in the Wapiti, it would be tempting to say that this very high-pitched vocalization is an adaptation to maybe a species colonizing an empty space, as the Wapitis colonized North America, for example, having crossed the Bering Strait. But it's how can we demonstrate this? And you've seen the variation. Most of these species can live in forests. They can also live in a semi-forested area. They can live in the tundra, and it's very difficult to. Yes, it's something we're studying at the moment with Elodie Briefer in Denmark, and many other colleagues. We're planning on doing propagation experiments and looking at what travels best in what habitat. However, what we're finding is it depends a lot on again on many factors, which are the height at which the animal gives the vocalization, the terrain, so many factors that there isn't a straight answer to this question. But maybe the best answer is, at the moment, I do not see any obvious mapping of frequencies onto specific types of habitats. There is no obvious, in the open terrain, open habitat, you have high pitch, whereas in forest habitat you have low pitch, or vice versa. We don't see that.
Sure. Okay. Thank you for that answer and for your talk. Of course, it was great to have you this morning. There have been more questions coming in, and I think as time goes by as well, we'll get more questions for plenaries as well, because these plenary sessions are available and people can post questions later on as well. So for speakers, we do ask you check back at your questions, and there's extra questions there that haven't been asked. We just haven't had the time this morning now. So please, encourage you to have a look at those and reply directly on the question and answer form. Thank you again, David.
Thank you very much.
Wonderful to have you.
For attending. Thank you. Have a lovely day. Bye-bye.
Cheers. Thank you. Bye.